set, let's get started with our first speaker, Lisa Barrett, Boston's own. Although I just learned that she's actually was originally from Toronto. Is that correct? So described as the most effective scientist in our time, the author of, among many other publications, the book How Emotions Are Made, which was acclaimed as brilliant and mind-blowing. And on one website, I found while Googling that it was described as her newest book, 7.5 Lessons on the Brain, was rated as the best beach read brain book. Lisa has just revealed that it was actually her husband who is the source of that quote. So if you are among the few who have not read Lisa's books, the time is now. And her talk title reminds us of the importance of the connections between our brain functions and our bodies, particularly the inner milieu of our bodies. So welcome, Lisa, and thank you.
Thank you. Thank you so much. Thank you for that lovely introduction. And I also want to thank Randy and Olaf and Tanya and everyone who was involved in the workshop for the honor of speaking with you today. I'll do a little bit of self-handicapping at the beginning, just to tell you that I'm actually recovering from spinal surgery, just, I've timed my medicine, so hopefully I will be lucid for the hour at least that I'm giving this talk. But I may be actually a little slower than usual, and that's going to be my excuse why for today. I want to say that in watching the talks, it seemed to me that I think it's a very laudable goal to give a talk for 15 minutes and then have the rest of the time for chatting. That's actually the best of all possible worlds, unless you're the speaker, and then you have real decisions to make. And so I've gone with the choice of showing very little data actually, and more just basically this is like a commercial for a set of ideas. And I assume that that we can get into the details, both the anatomical details and the empirical details as required by the questions and so on. So by design, this will be pretty just like and high level. And so I will start the talk. And so as Susan indicated, the goal of the talk is really to focus on the anatomy and on the intrinsic functional connectivity in the brain to justify the hypothesis that energy regulation, which we define as allostasis, and I'll define that further in the talk, as well as the sensory consequences of that regulation, which is called interoception, are at the core of brain architecture and brain function, and therefore may be at the core of all motor action and mental activity. And so I really want you to think about this not so much as in the context of verification or justification. I'm not trying to prove this hypothesis to you. I'm mainly presenting a really provocative hypothesis and trying to justify its value to the field. Because it's really not a hypothesis that any single scientist or any single laboratory can verify. And I'm not suggesting to you that everything can be reduced to energy regulation, but rather that a brain as a complex dynamical system, as we heard from Victor's talk and other talks, you can think of that as a system being built with neurons and other biological elements that continually interact via nonlinear dynamics, but that metabolism and energy regulation or regulation of the internal milieu may be overlooked as elements in the state space of that system. And that ultimately this hypothesis offers not only a principled approach for unifying very different phenomena into a single explanatory framework, but developing a shared vocabulary for theory building that will unite physical and psychological phenomena, both for health and for illness. And so at the outset, I want to start by thanking my lab, who you see depicted here, starting with Dr. Karen Quigley, who is a psychophysiologist, and she's the co-director of our lab. Dr. Yuta Katsumi who is a postdoctoral fellow in the lab, and Nada Kimona, who is a data analyst, and Donlei Chen, who is a graduate student, and also Dr. Jahud Zhang and Dr. Ian Kleckner, who are former members, and which is why you don't see them depicted here. I'm going to be talking about work that they've conducted in the lab, and in particular, a shout out to Yusa and Nada, because they really helped me pull the talk together, which on my own at this particular point would have been very challenging. So they helped me get some of the images together. And so the starting assumption for this talk and this whole idea really comes from evolutionary and developmental neuroscience. That the brain's most important job is regulating the systems of the body in the service of efficient energy regulation and action. And this idea is captured nicely by a quote from this really terrific book, Principles of Neural Design. The core task of all brains is to regulate the organism's internal milieu, meaning the internal systems of the body, by anticipating needs and preparing to satisfy those needs before they arise. The physiologist Peter Sterling refers to this as allostasis, which is not to be confused with homeostasis. And I'm happy to talk about the differences therein, maybe in the question and answer period. And as the brain is controlling the systems of the body, the autonomic nervous system, the immune system, the endocrine system, and so on, it's also modeling the sensory state of the body, which is called interoception. And interoception is not really your awareness of physical symptoms like your heart beating or sweating or your lungs expanding. Really, interoception is the brain's modeling of the sensory state of the body, that the sensory changes that arise from allostasis. To test whether there's any evidence in brain architecture that allostasis and interoception could be considered core functions of the brain, in our lab, we identified a set of cortical regions that in track tracing studies of macaques and other mammals have been verified to have connections to the brainstem nuclei that are known to be important for regulating the autonomic nervous system and the other systems of the body. So here I'm showing you just a sample of nodes because it's easy to fit on a single slide. And the thing to note about these is that they all have, their cytoarchitectural arrangement is either agranular or disgranular, meaning there's much less laminar development in these regions relative to other parts of the brain, other parts of the cortex, which have 6 very well-defined layers. And we're going to come back to this point because it's actually important, computationally important. Sometimes people refer to these as limbic regions in the sort of the old Paul Broca sense. And then we identified homologous locations in the human brain, and then using intrinsic connectivity analysis, we computed discovery maps, which were, as we've heard explained throughout the last couple of days, estimated as correlations in the time courses of low frequency bold signals when subjects are not being probed with an external stimulus, which is called resting state. And I think one thing to keep in mind about resting state is that even though we aren't presenting stimuli to subjects, sensory information is still hitting the sensory surfaces of the body. And in particular, the brain is still attached to the body. And so sense data, it's still controlling the body, and sense data are continually ascending to the brain. And this constant conversation is going on when the subject is at rest. And some of those signals, which are sometimes called intrinsic or sometimes called spontaneous cognition, actually are entrained by physical signals in the body. So we computed a set of discovery maps, and so we had a bunch of them, and then we performed a cluster analysis. And I should say everything that we did, we replicated on another set of subjects. So we had about 600 subjects in total. And so we did this cluster analysis, and rather than finding a single network, say, akin to what's been called there's something called the central autonomic network. We actually discovered two brain networks that summarize the connectivity to these limbic cortical regions, conventionally known as the default mode network and the salience network. Now, typically when these networks are depicted, they're depicted as topographically independent of one another, like in this image here from Yo et al. 2011. So as if they kind of fit together like puzzle pieces in the brain. But actually what we discovered is these two networks, when they emerge from this cluster analysis, they actually overlap in a set of hubs, some of which are members of the Rich Club, and so are some of the most connected regions in the brain. Now, these maps were derived from in a 3 Tesla environment, on approximately 600 subjects. We just replicated these findings on an individual subject by subject basis in a 7T environment with almost 100 subjects. And so the first thing to point out here is that these networks, whatever else they're doing, they also contain the key cortical nodes that are important for regulating the body. And these networks also represent the sensory consequences of that allostatic regulation or interoception. And what you can see here, I'm showing you the dorsal part of the posterior insula, which is actually primary interoceptive cortex. And so your brain is always regulating your body in every moment of your life, and therefore interoception is always occurring in every moment of your life. Now, most of the time you don't experience every tug of your chest, every contraction in your gut. You don't experience every temperature change and so on consciously, but your brain is certainly modeling them. Often what we experience is a low dimensional or highly compressed representation of those interoceptive changes as affect, as feeling pleasant or unpleasant, feeling calm or feeling worked up. And so you can think about these affective features as features of consciousness rather than as processes or anything like that. Both networks, so here I'm showing you the connections projected into the volume, brain volume. Both networks have more extensive subcortical projections than anything that anybody has discussed so far. And many of these subcortical nuclei are known to be important for controlling the autonomic nervous system and other systems of the body, such as the hypothalamus, the periaqueductal gray, the peribrachial nucleus, and so on. So the networks and their overlapping hubs have pervasive projections throughout an entire matrix of nuclei in the brainstem that are known to be important for allostasis. And not pictured here actually is the superior colliculus. And I was just telling a couple of people before the talk started that actually the superior colliculus is a key hub actually that is multimodal and involved in, receives interoceptive input and is also involved in allostasis. And I thought just for a second, I would also mention the mediodorsal thalamus based on Mike's talk yesterday. If you look at the anatomical literature on projections to the mediodorsal thalamus, you can see that it's receiving tremendous limbic input, not just from the amygdala and other subcortical regions, but also from the anterior cingulate and the ventral anterior insula and the posterior orbital frontal cortex, all of which are considered to be limbic cortical regions important for allostasis. So I think one thing that this should flag for you is that, we probably should stop referring to prefrontal cortex as if we probably should start referring specifically to where in prefrontal cortex we're talking about, because there are differences in the cytoarchitectural organization that that are relevant for understanding differences in the computation. And I should also say at this point too, that there are many, many, many experimental studies, both in humans and in non-human animals, that verify the involvement of these networks and their subcortical extents in the regulation of bodily systems. I just don't really have time to show those to you today. The two networks and their overlapping nodes also have connections to the hippocampus and the cerebellum. And I should point out here that these have been verified in track tracing studies, not just this. So what I'm showing you here is imaging data, obviously, but they've all been verified with track tracing studies. So both of these receive interoceptive information and are relevant for allostasis. There's a really interesting point about the hippocampus, which I won't go into in great detail, but theta oscillations, which are thought to be related to event segmentation and sequencing of events in the hippocampus, actually seem to have their origin in theta oscillations that are arising from the vagus nerve through the septal nucleus. So the medial septal nucleus. So the vagus is one of the main ascending nerve pathways bringing ascending sense data from the body to the brain. And so this suggests a really interesting hypothesis that event segmentation may be yoked to fluctuations in the physiological state of the body. Oh, that's just the septal nucleus. I mean, sorry, that's just the septal end of the hypothalamus. The septal end of the hippocampus. I'm showing you there. And here I'm showing you that brain regions that are in those two networks are also connecting to brainstem nuclei that originate the brain's neuromodulators and control the brain's attentional matrix. So these connectivity findings are augmented by an emerging body of experimental work suggesting that they may be performing domain general computations. So there's growing evidence, for example, that the behavioral state of an animal, meaning its allostatic condition and motor control, manifests as whole brain events and may even be involved in shaping perception. There are studies showing, for example, that the brain's sampling of visual information from the retina is linked to its modeling of heart rate, and somatosensory sampling is linked to respiration. There are studies showing that modulation of the autonomic nervous system occurs in standard cognitive tasks, like in our own lab. We saw modulation of the periaqueductal gray and other visceromotor regulation areas in the midbrain just during a standard NBAC task, with increased modulation as task difficulty went up or increased. So together with the anatomical work, these findings make plausible the hypothesis that the regulation of the body is an important part of whatever these networks are doing, psychologically speaking. So I'm not suggesting that the default mode network and the salience network maintain allostasis and represent interoception instead of all the other psychological things that they may be doing. I'm suggesting that it's happening simultaneously. That whatever else these networks are involved in psychologically speaking, and they've been linked to every domain of psychological function, at the same time, they may also may be regulating the systems of your body in the service of energy regulation. And so maybe instead of trying to look for one-to-one correspondences to demonstrate the validity of a network's function, a right approach might be something more domain general. So I have this talk timed to go to 20 minutes exactly. I think I'm probably going to go over, so I'm going to try to just speed up a little bit. I'm going to suggest that more specific hypotheses about what domain functions these networks actually serve can be found in the ever-growing literature on predictive processing, which offers a coherent neurobiologically inspired research program that can explain how the variety of cognitions and emotions and perceptions and actions can all be constructed using the same anatomical and neural features. So unlike psychology and psychiatry's traditional framework in which perception and cognition and emotion and action are in separate processes occurring in separate parts of the brain, each passing information or causing changes in the other, this approach suggests that psychological phenomena are created as part of the brain's internal model in its effort to navigate its body in the world in a metabolically efficient manner. Because the most efficient way to run a system is to model it and correct where necessary. And so we really have three elements that we need to be concerned about. So this has been discussed in Bayesian terms and with detailed equations and so on. And I'm just going to give you a gist level summary, which is a generative model that's constructed from past experiences, which are the prediction signals, incoming sense data from the body's sensory surfaces encoded as prediction errors, that is the differences between the predicted sensory inputs and those arriving from the sensory surfaces of the body, and precision signals that modulate the activity or of the patterns of prediction or prediction error signals. And there's predictive processing accounts of the cerebral cortex, of the cerebellum, of the hippocampus. I'm just going to focus on the cortex briefly today, because there's over 30 years of rich empirical work in primates and other mammals, which suggests that the arrangement of neurons within the cortical sheath predicts information flow across the cortex. So by looking at the architecture of the cortical connections, we can hypothesize something about the flow of prediction and prediction errors in the brain, again, in a way that highlights the importance of the body. And the general gist is this, that as signals, control signals, are originating in the deep layers of a less granular cortex, so less lamination and organization, as those descending signals go down to the thalamus and to the brainstem, collaterals are being sent to the upper layers of a more granular cortex. And these are prediction signals. And signals going in the other direction are prediction error signals. And so what this implies is that the cortical regions with the lowest laminar development, that is agranular and agranular cortices, will be at the top of the predictive hierarchy in the brain. And what are these cortical regions? They're the regions that are important for allostasis. So there's an irony here, and that is that for decades, scientists have considered limbic regions to be the home of emotions, which are supposed to be your inner beast, which is highly reactive and in need of control. But actually, the evidence suggests that limbic cortices give rise to prediction signals that drive perception and action in the brain. So prediction signals begin as visceromotor and allostatic control signals to the brainstem, and its efferent copies are sent to the more granular parts of cortex. That would be motor cortex, and then both sets of efferent copies from motor cortex and the visceromotor cortices are decompressed as they cascade to the sensory systems as sensory predictions. I was going to tell you about a cool experiment that provides very direct evidence for this, but there isn't really time. What I will say is just a shout out to Carl Friston, because when I first had this idea back in 2013 or 14, I can't remember exactly when it was, I wrote to him and we had a really lovely conversation, even though he didn't actually know me at all. He was very, very generous with me. So thanks for that, Carl. And I'll just say more recently, our lab and other labs have been taking a more gradient approach to see if we can understand the role of allostasis, and we are using this for the purposes of understanding the role of allostasis and interoception in parsimonious organizing principles that describe structure-function relationships as low-dimensional connectivity gradients. And so in our first paper, we took 109 cortical nodes in the brain that anchored the various intrinsic networks that have been published in the literature. And we created functional connectivity maps for each. Here I'm just showing you the cortical parts, but actually they were whole brain maps. We computed the similarity between them and thresholded it, and then did a dimensionality reduction. And so as we were preparing this for publication, actually Daniel Margulis came out with his wonderful paper in PNAS. Luckily, we replicated him in very important ways. So what I'm going to show you here is a multidimensional scaling analysis, which is what we used. And then in the replication, which I'll just show you near the end, we did a voxel-wise similarity of intrinsic functional connectivity patterns using diffusion map embedding. And so here's a plot of the two main dimensions of map-based similarity. Each point on the scatter plot represents a profile of connectivity for a particular node in the cortex, and the colors indicate the network that the node is usually assigned to. And the first thing that you see here is you don't see clusters, you see actually what is called a circumplex anchored by two smooth gradients in Euclidean space. The first gradient is actually, we think, represents a predictive hierarchy where there are a number of architectural hierarchies, or I should say architectural gradients in the cortex, which align with this particular dimension. And the way that we understand it is that default mode network may be the home of the brain's internal model, with the executive network or frontoparietal network really really modulating the internal model, that is the precision of the predictions. Whereas at the other end of this dimension, you have information coming from the sensory periphery and the networks that are modulating or estimating the precision of that prediction error signal. And then this second dimension is really a precision versus model, where the three networks at the top, executive or frontoparietal, control, salience, and attention, sometimes actually cohere together, as the test positive or multi-use. So they're modulatory networks as opposed to content representing networks, which are the bottom two. And I'm going to just whip through this just so that you can see really quickly that these gradients, actually, you can see them in the cerebellum and in the hippocampus. And that's actually true for both gradients. So my summary is that my hypothesis really in the work we do in the lab is that really the default mode is the origin of prediction signals along with the hippocampus and the cerebellum, and its precision estimates are being computed by the frontoparietal control. And we can talk about that in terms of decision-making or planning and so on. That the salience network is important for precision of prediction errors, and that's consistent with its role as a multimodal integration network. And the hubs are important for integration and synchrony in the way that Olaf Sporns and Martin van den Heuvel and so on has shown. And I'm not just gonna thank you very much for your attention and your patience, and I can take questions.
All right, thank you very much, Lisa. Well, we have had a few questions come in through the Q&A, and so why don't we begin with one of those. And in the meantime, people who are on the panel, please, raise your hand, and you have the ability to unmute yourself when you want to ask a question. But let's start with a question that was asked by Andrew Lynn. Andrew, if you're on the call, if you're on the Zoom, do you want to unmute and raise your hand so we'll see you? You can be unmuted, and we'd like you to ask your question. No? All right, Andrew, I'll ask your question because it's an interesting one. So Andrew asked, one concern that commonly comes up with resting state connectivity data is controlling for physiological signals, such as heart rate, respiratory rate. Could changes in these signals track allostatic changes and the dynamics of the networks you've characterized?
Sure, and we control for that, so I should have said that. But I think there are two ways to think about this, right? So one is that, for example, blood flow changes that occur due to like subtle blood flow changes that occur due to changes in heart rate or changes in cardiac output, you would consider that to be a confound in the bold signal. But if you do something like you can do, you can use electrical signals to compare I'm trying to think of the word and unfortunately I can't think of it, but you can look for signals in the brain that are corresponding to respiratory oscillations or gut oscillations or heart beat changes that are not due to, that you wouldn't think of as a confound. And once I think of the name of the task, I will tell you. This is, remember I said I'm on pretty heavy medication for back surgery, so recovery. So this is me on medication. And in any case, those, you wouldn't think of them as confounds. You would think of them as actual signal, not noise. So, and I should also point out to you that almost all of the connections that we show in our imaging findings, we also verify in track tracing studies. So we always anchor everything to track tracing studies when we can find them. And so, for example, my point about prefrontal cortex, it's very misleading to refer to prefrontal cortex because there are aspects of prefrontal cortex which have very dense connections to the hypothalamus and the PAG and so on, like anything in the cingulate and anything from ventral medial prefrontal cortex all the way up through dorsal medial and even parts of ventral lateral prefrontal cortex. The places where you don't see connectivity really in track tracing is in dorsolateral prefrontal cortex. And so even if it was the case that we did not control for the artifactual aspects of those signals, I would still point out that the track tracing evidence is pretty clear. And there's also experimental evidence. So the experimental evidence is sometimes from bold signature, from bold data, but not always. Oftentimes it's electrical or humoral.
Great, thank you. Thanks for that question and thanks for that answer, Lisa. Our next question is also from one of our participants out in the audience, and it's Debbie Yee. Debbie, would you raise your hand so we could be unmuted so you can ask your question? And if you're not able to, I'll ask your question for you, but I'll give you a second.
Can you hear me?
Yes, thank you.
Great. This was a wonderful talk. I just had a question, maybe to summarize briefly about the insula. I just noticed that you kind of made some claims about posterior insula, but looking at these maps, there seems to be kind of a broad coverage of insula. And I was wondering if you had any thoughts about whether there might be functional or anatomical dissociations or like maybe a spectrum that might help us understand, like maybe different subfunctions of allostasis or whatnot that might kind of inform some of the predictions.
Yeah, so I certainly am not advocating for treating any swath of cortex, as anything uniform, including the insula. So there is evidence both from track tracing studies and from imaging and from electrical stimulation studies that the mid to posterior dorsal aspect of the insula is primary interoceptive cortex. So I can point you to a number of papers which summarize that evidence. But of course, the insula, the more anterior parts of the insula also contain primary gustatory cortex, primary olfactory cortex, and there's actually a lamination gradient in the insula. For example, posterior insula is granular, so it's sensory in its organization. But the more anterior you go, and also the more anterior you go, the more granular, the more agranular you get, like the less granular the cortex. And also, as you go from dorsal to ventral, you also get less granular. So, for example, ventral anterior insula is equivalent to what some anatomists like Helen Barbas call posterior orbital frontal cortex. It's actually the same part of the cortex. It's agranular, and it's part of Unger and Price's, visceral motor network. So it has very, very heavy projections to the subcortical regions that I was mentioning, even though it is also serves a function as an integration area, sensory integration area. So one thing I didn't really have a chance to talk about today is that one of the interesting things about this cytoarchitectural gradient, which I've been talking about in terms of lamination, it's really a compression gradient or a dimensionality reduction gradient. That you're going from a lot of little neurons with very few connections all the way to, the information is percolating or it's being compressed in a lossy way to, neurons that are bigger, fewer, but with many more connections. And so you can think about these cytoarchitectural gradients as compression gradients, where you're going from very high dimensional sensory detail to lower dimensional representations. Bud Craig talked about the insula, the posterior part of the insula as being more like a sensory, like primary sensory area, interoceptive, and the anterior insula as being more about re-representing these signals as affective or emotional. And I don't think that's the right way to think about it. But there's the gist way in which he's sort of correct, but I wouldn't call it re-representing, I would call it dimensionality reduction. What's happening is the information's being compressed. And I thought Mike yesterday gave a really nice definition of compression. I would say there are compression gradients kind of all over the brain, really. If we had time to talk about it, we could talk about compression gradients in the hippocampus and in the cerebellum and even in the basal ganglia. And so I would say, yeah, I think that there are more specialized computations being performed in different parts of the insula, but I don't think for example, like I said, the front part, you can think about it as having compressed sensory representations, but it's also visceral motor, and it has direct projections to the PAG and the hypothalamus and so on. And it's also part of the multimodal multisensory integration network that's responsible for sensory integration, which, by the way, is also a compression type, dimensionality reduction type computation.
Thank you. Thank you, Debbie, for that question, and Lisa for your rather extended answer. We appreciate that. I'm going to turn now to our panel. So Randy McIntosh has patiently had his hand up. So why don't you go ahead, Randy?
Sure. Thanks, Lisa. That was fantastic. Even though you're on drugs, you're still pretty good. So.
Until I remember the name of that task, it's going to drive me absolutely insane. But please, yes.
I got a sort of a philosophical question. This actually comes from sort of reminiscing about my early days in neuroimaging when I was talking with people like Endel Tulving and so on about using neuroimaging to actually understand sort of the essence of human thought and coming up with ways to actually measure the brain so that we would be able to extract sort of the core of our being, if you will, and get rid of all the other fluffy stuff on the outside. If I understood sort of the message of your talk, and also in some respects the message in your latest book, that's maybe almost not a wise endeavor because the brain is so intimately intertwined with the body and the environment. And I get the impression that if we're really going to make headway in terms of understanding, as you say, the cognitive functions that are actually helping us to integrate the brain and body, we actually have to try and research them together as opposed to making a sharp boundary between, cortex, thalamus, cerebellum, brainstem, and everything below. Would that be accurate, or am I kind of missing something there?
No, I think that's accurate. And I think there's a danger in saying, oh, do you think we should study everything, all at the same time? And I'm certainly not saying that. But what I am saying is, if I'm not going to give you a whole lecture in brain evolution, but I will say that it was a real surprise to me to learn that the earliest creatures and the simplest creatures have motor system and virtually no exteroceptive sensory systems at all. I mean, they have very rudimentary somatosensory. It's not a lateral line system. It's a very, very rudimentary somatosensory system. No eye, no visual system. A light, a light spot for circadian regulation, and so on and so forth. And so, if you look at both the evolution of the nervous system and also if you look at embryological development, so how things develop off the neural crest in an embryo, what you can see is that exactly in the same parts of the neural crest, you get a head, you get sensory systems, you get the forebrain and visceral systems. And they're all coming from the same parts of the neural crest. And they all evolved approximately along the span of evolutionary time around the same time. And so I guess what I would say is from that perspective, but also just from a functional perspective, your brain's, most important job is regulating your body. I mean, because if it doesn't, you're dead. And there are really fundamental ways in which that's true and can be misleading if we don't consider that. So for example, cortisol is not a stress hormone. That is the biggest misunderstanding that stems from trying to separate, cognitive things and emotional things from physical things. So cortisol is a glucocorticoid that is a hormone that gets glucose into your bloodstream quickly when your brain predicts that you have a big energy expenditure that you have to make. And that's all stress is. Stress is your brain preparing you for a big metabolic outlay. Now, you have a cortisol surge right before you get out of bed in the morning. You have a cortisol surge right before you start exercising. You have a cortisol surge right before you try to learn something really hard or do something hard, like an NBAC, with with a load of 3 items. All of these things are expensive and difficult, and your brain prepares for that by releasing cortisol. And actually, there are direct connections, like you can trace, I think it's just a couple of synaptic links between the anterior cingulate, for example, and the adrenal glands. This is really nice work by Peter Strick. So I think that particularly for human neuroscience, where we, now we're moving to much larger sample sizes, but, for many years, much smaller sample sizes, where people can come in at any time of the day, in any metabolic condition. This is all meaningful variance that is uncontrolled in very small samples and probably wreaks havoc. I think one of your colleagues actually found that even performance on cognitive tasks can shift by almost a standard deviation, depending on whether you bring someone in at the peak time of their circadian rhythm or at an off time of their circadian
That's from
rhythm.
Lynn Hasher, yeah.
Yeah, exactly, Lynn Hasher. So yes, I don't think everything can be reduced to the physical functions of the body, but I do think that this is the best way to say it is that it's really a part of the state space that we have ignored. And even yesterday, when you asked, Randy, you asked this wonderful question, but I like you said, you asked, I think it was after Bia's talk, you said, well, how do we separate motor preparation from cognition? And I was thinking, I don't think they're different. I mean, I actually think part of what we think of as cognition is actually the brain deciding, as it were, doing decision about which representation is going to have access to the motor systems. And if you look, for example, the anterior cingulate, the parts that are called cognitive are actually motor. So I think that there's really, from a philosophical standpoint, a really deep point here that we have so far been overlooking, mostly because we're unaware actually of the whole drama going on inside our own bodies most of the time.
Thank you, Lisa. In fact, I wish we had unlimited time to discuss your last point, because I think it's really getting at the heart of what the brain connectivity workshop is really aiming at, all the way back to the very beginning, Randy, right? When we asked, what does the brain think of the mind? So
Yeah, definitely.
I'm going to take one more question from the panel. Carl has also been quite patient. And in fact, as he's recovering from his COVID vaccination, we're gonna give him a treat, and then we'll return and get some interesting questions from the attendees that have been posting questions. So go ahead, Carl.
Yeah, I'm still recovering. I think vaccination for coronavirus stops your cortisol surge in the morning. I'm still waiting for mine. But I was invigorated by the talk, so that was a bonus prize. Before I ask my question, which is actually a follow-on exactly from Randy's and Susan's observations, I just can't resist saying how much I like this notion of gradients of compressibility as evinced by the neuroanatomy. And that speaks so directly to the computational architectures of things like variation autoencoders, information bottleneck. And also, interestingly, the very first predictive coding algorithms were invented in the 1950s to compress sound files. So the whole, if you like, ideology behind predictive processing started exactly with this notion of compressibility. So I thought it was very attractive. The other thing I liked was this sort of dual gradient of predictions of content and predictions of precision. And in that setting, your slightly controversial but compelling argument that the deepest, most central, highest aspects of our hierarchical anatomy, in fact, the oldest sort of limbic and paralimbic bits of the brain, which is slightly counterintuitive, but I'm sure you're absolutely right there. Even to the extent that you might make friends with a chap called Mark Soames. Have you come across Mark Soames?
Yes, I have. And I would say there are some deep divisions, however, because he really comes from this Panksepian tradition, believing to speak of philosophy, he, I think, confuses circuitry for actions with circuitry for emotion. And that's a whole other debate to discuss. But basically, there's a tendency in neuroscience and in psychology to define fear as freezing behavior. And therefore, when you find the circuits for freezing behavior, you have found the circuits for fear. And I think there's hugely problematic, because there's tremendous diversity in what animals, how animals deal with predators. And in a human, so I'll say, humans do many things in fear, and therefore the circuits that will be the sort of circuits that are lurking in subcortical anatomy, those circuits, there isn't a one-to-one correspondence between any behavioral circuit and any emotion category. And I'm happy to unpack that, but it's a longer conversation. So I wrote a paper with Barb Finley, actually, where I was sort of addressing this issue, and also the issue of survival circuits, how Joe Ledoux's kind of revision of his understanding of his fear-related work, and or initially what he called fear-related work. And I guess what I would say is, so I think that if Mark would give up the devotion to aligning emotion categories with circuits for actions, then I think we would have a lot more to talk about. So I'm very much interested in understanding ancient circuits for behavior, but I don't necessarily think the evidence suggests that they map onto human mental categories in a one-to-one way, if that makes sense.
No, absolutely. I was asking, in a slightly more practical way, because of his recent focus on the subcortex and the ascending classical neuromodulatory systems, the dopamine, the serotonin, and his most recent story that consciousness lives in those neuromodulators and it's not even in the cortex, which is even more radical than your assertion.
But I was talking earlier before the session started about, for example, Bjorn Merker has written, beautifully, I think, about the superior colliculus as a potential important area that is important for consciousness because it's a major integrator, multimodal, it's a multisensory integrator, and it's also, in integrating internal with external signals. And so, I tend to think that there isn't going to be one place or one set of chemicals that we can attribute to consciousness. That really what we are probably talking about is, a complex system, and the state space for that, or the state vector for that, the state variables for that system are probably numerous. So I'm skeptical of simple one-to-one. Dopamine and serotonin and norepinephrine all evolved as metabolic regulators. And so, there's a whole conversation to be had there about what dopamine actually does, just from a metabolic standpoint, without layering on all the psychological stuff that we usually do.
Thank you. So it's not the pineal gland after all, then. That's good to know.
I tend towards complexity, I guess. It's just, maybe that's just my penchant. By the way, heartbeat evoked potentials. That's the task that there are evoked potentials from the heart, from the gut, from the lungs, and so on, and you can track them electrically in the brain. And so it doesn't rely at all on blood flow or bold signal or anything like that.
Great. Thank you, Lisa. And thanks for stimulating that interesting conversation, Carl. We're going to turn now to a question from one of the attendees. Lorenzo Pasquini, are you there? Yep, I see you. Great. Go ahead and ask your question, please.
Hey, hello. Do you hear me well?
Yes.
Thanks. Lisa, thank you so much for this very intriguing talk. If I understand well, some aspects of the allostatic interoceptive model predicts that emotional states are true thing, that they result from a match between incoming signals from the body and from the sensor, the visual system, for example, and how they match into the predictor generators. And depending on what's coming from the body and in which environment we are, this match will generate distinct emotional states. And you will correct me if I'm imprecise on this, but that's my basic understanding of the model. What I don't understand very well is, why is this model, if I describe it correctly, suggesting that there are not specific patterns that can predict with a certain probability what is the underlying emotional state coming from the body and from the brain. Well, I understand that this can't be deterministic. What I don't understand is why there can be still some pattern in the brain physiology or in the bodily physiology that can, give us a hint.
Well, thank you for that question. I never said that there's no predictability or no patterns. What I said is there isn't a single pattern. So a category like anger or sadness or fear is a population of instances. There isn't a single pattern that predicts anger or sadness or fear. And I can show this to you in physiology. I can show it to you in brain data, in electrical stimulation data, in bold data, and so on. It would take longer than the 2 minutes we have left for me to go over all of that evidence. But what I think is really interesting, Lorenzo, is that oftentimes when I say there's no single pattern, meaning that we really have to think about the internal context and the external context and the action that the animal is preparing to execute. We have to understand those things in order to, because the pattern of activity is really linked to the action that the animal is going to take. And that there isn't a single pattern, you don't do one thing in anger, you can do many things. And the pattern will be dictated or will vary as the movements vary. So, but when I say there's no one pattern, people take me to mean that there is no predictability. And I'm not saying there's no predictability. I'm saying there's a distribution of patterns. And if there was more time, I would link the idea of, or I have linked the idea of gradients of compressibility to understanding this distribution of patterns. And there's a way to understand this actually as category construction or concept construction, that really what a brain is doing is when it constructs a prediction, it's actually constructing a distribution of predictions, you could say, or a population of predictions or sample. It's constructing a category, but it's an ad hoc, context-specific category. So you can predict instances, but you can't say that any, there's a reason why multivoxel pattern analysis in one study doesn't, it might distinguish anger and sadness and fear, but it can't predict, subjects or instances outside the sample. And it doesn't look anything like some other pattern, which in another study also distinguishes these instances of these categories, but the patterns look nothing alike. And the answer is because they're sampling patterns because the actions that are required are different. And the
Thank you.
physical state of the subject may be different too. We're not controlling for it or measuring it in any way.
Thanks for helping me understand this better.
Sure.
Thank you. Thank you, Lorenzo. Thank you, Lisa. Okay, we have a question from an anonymous attendee that I thought might be we could pose to you, Lisa. He says, he or she says, what is your thought on OFC being functionally active in a lot of cognitive tasks, including decision-making, attention, and emotion, in spite of its not so direct involvement with allostasis?
Well, first of all, it has, I think OFC is a really big swath of territory. And so if you look, for example, at the Unger and Price maps, these are papers from, let's say, 2003, maybe 2003, and also in the 1990s. It's very clear that there are from their perspective, two systems really in orbital frontal cortex. The lateral parts of orbital frontal cortex are really more multi-sensory integration regions, and we might think of them as being, on that compression hierarchy. And then the more medial, posterior and more medial parts are actually directly visceromotor. So they're very involved in allostasis. In every animal that they've studied, which I think in most of those papers are mostly rodents, monkeys, and humans. And I think, although we don't tend to do this, I think we also have to think about we think about, for example, the insula being very, very separate from orbital frontal cortex, but the most posterior parts of orbital frontal are actually are the more anterior parts of the insula. And just in the same way that the most ventral parts of ventral medial prefrontal cortex and ventral lateral prefrontal cortex are also considered orbital frontal cortex. So I guess I would maybe disagree with you that OFC is not involved in allostasis. I think the evidence is that certain parts of it actually are, reliably enough that these authors would make claims about there being fairly stable computational differences in that cortical swath of cortical area.
Great. Thank you, Lisa. I think we're going to have to make that the last question. I think you really have challenged us to rethink a lot of the perceived wisdom that we kind of have about the way we carve up both the brain, the body, and the mind. And so I think you've certainly created a lot of work for a lot of people to do over the next decades, thinking about their own work in this new framework that you're putting out. So thank you very much.